Thank you for giving us this opportunity to share our findings from our theme-based research program. And at the outset, I would really like to express our heartfelt thanks to the Research Grants Council for funding this program. You can see some of the co-investigators, co-principal investigators of this program. And the project spans 3 universities, and multiple faculties, not just the faculties of medicine at HKU and CUHK, but also faculties of engineering and faculties of science. The objectives and goals of our program was to understand transmission of viruses, respiratory viruses, influenza being the example, and also to understand how these viruses cause severe disease in the lungs. And then finally, to develop evidence-based interventions to stop or reduce transmission and to prevent this pathology. And in addition to that, to foster the development of the next generation of researchers. Now, the program was focused on influenza, but as you know, at the beginning of this year, we had another respiratory virus and coronavirus. And as I will show you, the expertise that we had developed in this program was very efficiently transferred to tackle the challenge faced by COVID-19. So we have quite a number of research publications coming out from our theme-based grant, over 150 and still counting. Already cited over 3,000 times and published in the best scientific journals, Science, Nature, Lancet, etc. And I won't, of course, have time to cover many of these. And my apologies at the outset to my collaborators whose work I will not be mentioning in this short 15 minutes presentation. I also want to highlight some other achievements of the theme-based program. We organized 2 international conferences, one on respiratory virus transmission, and another Keystone conference more recently. So let me just highlight a few of the achievements. So one of the big questions we were trying to tackle is how influenza and other respiratory viruses transmit from one person to another. So the dogmas were that the transmission takes place by these large droplets that fall to the ground within a meter or 2, small airborne particles that can float in the air for much longer distances, and also indirect transmission through contaminated surfaces and hands and transfer to the respiratory tract. But a lot of these really did not have a scientific foundation for these recommendations. And this we identified as a major knowledge gap. And that was one of the major reasons for our study. So I mentioned this international congress that we organized in Hong Kong. And within that, we had a smaller group of people who got together to try to address one specific issue. That is the experimental models, the experimental animal models that we use globally to investigate transmission of influenza, which is the ferret. As part of this meeting, we had a sub-meeting where we discussed the variables because many different labs use this in different ways. And following on from that, we had an international group of people, laboratories from the USA, the Netherlands, UK, Australia, and of course, Hong Kong, who are using these technologies, where we came to try to harmonize the methods that we use and also use some evaluations to see how uniform our results are. And this was co-led from Dr. Yen from the theme-based research program and Dr. Jessica Belzer from USCDC. Now, again, using this ferret model, we were able for the first time to develop an innovative method to try to quantify and assess what sized particles, airborne particles, transmit influenza in the animal model. I won't go into the technical details of this, but this was possible with the collaboration of the Faculty of Engineering, who are experts in aerobiology, and of course also in the fabrication of some of these equipment. So we were able to show that in the ferret model for the first time that the transmission efficiency correlated with the large airborne droplets larger than 1.5 micrometers. And very interestingly, we saw that the transmissibility declines over time, even though these airborne droplets continue to be shed. So this led us to conclude that there are host factors in addition to the shedding of these infectious droplets in transmission. So this is, again, a very unexpected and novel finding, which we are continuing to investigate. And now this whole approach is now being transferred to humans to investigate the infectious particles that humans exhale from their breath. Now, we transferred this expertise in airborne particle measurements and detection to the field. And one example is live poultry markets. As you know, avian influenza is a big challenge in our region. And we were able to show using these technologies that you do have significant amounts of infectious airborne particles carrying influenza viruses in these live poultry markets, and that the main source of the generation of these is this defeathering machine, which is what is used once the chicken is killed and the carcass is put into this machine, which spins at very high speed to remove the feathers. So we were able to show that this is one of the major sources of these airborne particles. And again, in collaboration with colleagues in the Faculty of Engineering, who again studied the aerodynamics of this situation, they were able to design a modification to this defeathering machine, which markedly reduces the dissemination of these airborne particles. Now, as part of all these studies we were doing, we have continued to be providing advice to the Department of Food and Environmental Hygiene of Hong Kong in how to improve the hygiene and reduce risks of avian influenza in the live poultry markets of Hong Kong. We also have been, and this is studies led by Ben Cowling, looking at the release of airborne particles from humans who are infected with influenza was the primary aim of the study. And this was published earlier this year in Nature Medicine. But what was particularly topical, given the emergence of COVID, was that some of the patients that we recruited to the study did not have influenza. They had coronavirus infections, not COVID-19, because this was done before this year, but other coronaviruses. And there was no data on coronaviruses of this nature before. So what you can see here is that we were able to show that patients with human coronavirus infections do release large droplets and small droplets, small airborne particles that are potentially infectious. But even more important, we were able to show that when these people wear surgical masks, the release of these droplets, both the large and the fine airborne particles, were prevented. So essentially, the wearing of surgical masks not only protects the person who's wearing it from infection, it also prevents people who are infectious from transmitting to other people. And taken together with the realization that in COVID-19, people are infectious even before they develop symptoms, this was one of the underpinnings for the recommendation that for the universal wearing of surgical masks in the community. And as you can imagine, this was very popular paper. It has been downloaded more than 2 million times, and it was one of the most heavily downloaded papers from the Nature journal system in the first month or so. And it's already been cited over 300 times, although just published only a few months ago. Another area of interest was transmission within households. So essentially, again, studies led by Ben Cowling was to identify patients in a household who are infected with influenza, and then follow up the other contacts in the household to see when they get infected, who gets infected, why people get infected, so that we get very early data on infection and infectiousness. And there has been a whole series of important publications coming out of this work, but just to point out one, again, able to show that influenza is infectious prior to symptom onset, and this has, of course, implications for control. And also that influenza A and influenza B behave quite differently in terms of their transmission dynamics and viral load kinetics. In terms of preventing transmission using vaccines, again, studies were able to show that the duration of the effectiveness of influenza vaccines is relatively short-lived. Of course, we know that influenza vaccines have to be updated because the virus mutates, but what in addition we were able to show, because of Hong Kong's long seasonality with flu, is that even with the same virus, even if it's not mutating, the duration of protection declines. And understanding this is important for improving vaccines in the future. Another big question we were aiming to tackle was how and why some of these respiratory viruses cause very severe and fatal lung disease. And the example we were using as our model was avian influenza H5N1. So my colleague, Michael Chan, was able to set up an experimental model to study this in the laboratory. And what I'm showing here is one of the air sacs in the lung, and we have millions of these air sacs in a healthy lung. And normally, the fluid that leaks out into these air sacs is pumped out by a system of transporters, ion transporters, chloride and sodium transporters. But what Michael was able to show was that when a virus like H5N1 infects these lung epithelial cells, it blocks these transporters. And this results in accumulation of fluid in the lung, which is what leads to the severe disease. And he went on to show one intervention which can reverse this. And this is a mesenchymal stromal cell treatment in this model, but also in a mouse model, that it can reverse this severe lung damage. So this has a quite significant potential for therapy. And we are now aiming to use this experimental model to look at other small molecules that might be able to do this. And this will be part of a Hong Kong program. Given the expertise on respiratory viruses that was developed as part of the theme-based program, when COVID-19 hit, we were well placed to very rapidly develop diagnostics. And indeed, within days, so by the 24th of January, were able to validate and post a diagnostic method on the WHO website. This was one of only 2 methods available at that time globally for diagnosing COVID-19. And given the huge demand from many countries for diagnostic reagents, we supplied over 170 laboratories in 77 countries in those very early days when commercial diagnostic kits were not available. Again, transferring our expertise from the theme-based grant on animal models for influenza transmission, my colleague, Dr. Yen Huiling, was able to show the hamster to be an excellent model for disease and for transmission in the case of COVID-19 or SARS coronavirus type 2. So what you can see here is infected cells of the lung epithelium. You can see the nasal epithelium and the olfactory sensory neurons getting infected, which explains why patients with COVID-19 lose their sense of smell. And as I mentioned, also showing that you can study transmission from one animal to another using this experimental model. So this again is a very heavily downloaded paper, already cited over 80 times, again, just within a few months of publication. So just to highlight the international role and international recognition our group has as a result of the theme-based grant. We are a WHO collaborating center for infectious disease epidemiology, headed by Professor Cowling. We have 2 WHO reference labs for influenza and for COVID-19. We participate in regular WHO meetings for influenza vaccine strain selection. We have contribute heavily to WHO FAO activities, including producing consensus papers and guidelines. So this again on non-pharmaceutical interventions, so that is public health measures that can be used to contain the spread of influenza, was an important contribution led by Professor Cowley, which now has been an important foundation for control of COVID-19. And we are board members of important respiratory disease bodies and journals, and also serve as scientific members of scientific advisory boards to some of the major research institutes in the world. We have trained over 50 research postgraduate students and a number of postdoctoral fellows and research assistant professors, and 8 of our postdoctoral fellow research assistant professors now have secured tenure track positions in Hong Kong universities, and one, for example, was appointed as associate professor at the University of Oxford. We are very proud of our knowledge exchange program, where we have an outreach program called Little Dr. Flu, where we bring in students from primary school to teach them the basis of infectious diseases. And we have, of course, many international collaborations, but just one I want to highlight is this collaboration established with Dr. Jungnan San's State Key Lab for Respiratory Disease in Guangzhou. And this has been extremely productive. We already have a Greater Bay Area collaborative project with them. And during COVID-19, we were able to do a lot of very important collaborative work together with them. So just to highlight the fact that we have, I think, achieved all the key objectives that we set out at the start of the program. And in terms of the continuation and the legacy of this program, we have a follow-up TRS, theme-based program led by Professor Cowling now. We are investigators on US NIH, NIAID-funded Centers for Excellence of Influenza Research and Surveillance. We have 2 HMRF commission studies, large studies on COVID-19. As I mentioned, recognized global leadership in the area. And also, as I mentioned, we have a Hong Kong project, which will extend some of the findings we have into translation and commercialization. So with that, I will end, and I will be happy to take any questions. Thank you.
Maybe I have one question first. What do you think are the most urgent priorities now in COVID-19 in terms, what do we still need to know that we don't yet know?
Yes, I mean, I think what COVID-19 has shown us is the vulnerability human societies, economies, and the world in general has to a highly transmissible respiratory disease. And this could be influenza, it could be COVID-19. Now, another thing is that, again, what it has highlighted is that in the early phases of such a new emerging infection, we don't have vaccines. We may not have antivirals, as was the case for COVID-19. So the only thing we do have are the public health measures. And so really understanding the scientific basis for how these viruses are transmitted is fundamentally important. Because, for up to now, really, the control measures for COVID-19 has been physical distancing, wearing of masks, and reducing mobility and things like that. So, and question of closing schools, is this useful, is it not? So these are really very fundamental questions to which we need a scientific basis. And I think we can be proud that this theme-based program has contributed some of that fundamental knowledge to flu and to COVID-19. So that is one big area which we still need to continue to push forward. Then, of course, there are other issues. I mean, the question of immunity. How long does immunity to COVID-19 last? We know that many people do develop good antibody responses. And how long does that last? Does it wane? Does it disappear? What are the correlates of protection? Because having antibodies, there are many viral proteins to which antibodies are raised, but what exactly are the protective determinants and what are the titers that are protective? This again remains to be seen. And this again will become fundamentally important as we roll out vaccines. Then another question, it's really very heartening that vaccines are now being licensed, which seem to be quite effective. But we still don't know whether these vaccines, although they are preventing disease, whether they will prevent transmission. A lot of people assume that the two are the same, but as you well know, Ben, they are not the same. So we need to know whether or not these vaccines prevent transmission and what types of vaccines do that job better. So I think there's a whole host of questions And in addition to that, of course, the question of how COVID-19 causes lung disease and how we can reverse it. So that comes back to some of these studies with H5N1. So I think a lot of things that we can address directly coming out of the theme-based research program.
On the topic of vaccines not preventing transmission, I guess the problem would then be that we may never quite reach herd immunity in the same way as we might do with other diseases and other vaccines. So COVID would always be a threat, maybe in vaccinated people. It wouldn't cause much illness to be concerned about, but there would always be unvaccinated people around in the population, or if the vaccine effect wanes over time, there would always be people whose vaccines have worn off or worn out or not working. And then that could pose a real problem going forwards, even in the post-vaccine era. And I'm not sure how, what the best way is to deal with that.
Yeah, so I think first and foremost, we need to establish the extent or presence or lack of prevention of transmission with the existing vaccines. And that is the foundation to see, how we can develop the second generation of vaccines, which may be better at preventing transmission as well. I mean, I think I just want to emphasize that I'm not how shall I say, reducing the huge impact that vaccines are going to have. Whether or not they prevent transmission, COVID-19 vaccines, by protecting, hopefully, the most vulnerable, will have a huge impact. But as you rightly say, in the longer term, what we also need is to prevent transmission, the same way that, for example, the measles vaccine prevents transmission in the community. And so this, I think, is still an open question. And if it is the case that the current vaccines are not so effective at preventing transmission, that will point us in the direction of the gaps we need to fill. And theoretically, one could imagine that intranasally administered vaccines, because they provide local immunity in the nasopharynx, might be much better at doing that. And maybe we may have to have two types of vaccines, or maybe ideally a single vaccine will do both. So I think we are still at the beginning of the journey in regard to tackling COVID-19. We have come a long way in the 10 or 11 months, but I think there's a long way to go, and there's a lot of research left to be done.
Do you know much about long COVID and what might be the underlying mechanisms or the reasons for why this minority of people after recovery still have some kind of symptoms in the longer term?
Yes, again, that's an important question, because I think, as you rightly said, I don't have the answer to that. But I think this highlights a very important issue. And I mean, a lot of people assume that younger people, they don't develop severe disease, and therefore we don't have to worry too much, and they can just go out and get infected. But it appears that young people can develop very mild disease, but can still develop the long-term complications of COVID, which is called long COVID. So this again has implications for our control measures. And of course, presumably this is because of, we know that the virus actually does persist for weeks and for months. It is not infectious, but it does persist. I mean, there are recent studies showing the evidence of viral replication in the gastrointestinal tract, I think 2 months after onset of symptoms. So there's a lot more to be learned about the persistence of the virus, but then also the host response that is generated, the innate immune dysregulation that the virus elicits, and the consequences of that. So again, I think all these are questions, I think, that the team, this theme-based research program, and indeed the next talk we are going to hear about, I think these theme-based research programs really have assembled a global leadership team in Hong Kong. And I think it really would be great if the RGC continues to fund these theme-based programs, because I think they are really value for money if you think about the totality of the output. Thank you.